Hi everyone, my name is Carmen Reep, and together with my supervisors Katie, Eleni, and Nuria, I created an automated drug repurposing for rare diseases. And during this presentation, I will tell you how we did this. Oh. So let's say we have this patient who suffers from memory loss, has involuntary unpredictable muscle movements, and has behavioral problems. This patient is diagnosed with Huntington disease. This is a rare neurodegenerative disorder. Only 1 in 10,000 people suffer from this disease, and there are currently no effective treatment options. So how to help this patient? Ideally, you would find a new drug, but this is a very time-consuming and costly process. So pharmaceutical companies do not see benefit in developing a new drug as they don't have profits if only a few people suffer from this disease. So it would be better to find an already existing drug to help alleviate the symptoms of this patient. And this is called drug repurposing, and this is what we've done in this project. There are many different drug repurposing approaches, and we use a computational approach, which means like the analysis of any type of data. Computational drug repurposing has been done before, but it still had some challenges. One challenge is data access and integration. And this is where our contribution comes to the field. So we use the Bio-Knowledge Reviewer library, which was built by my supervisor, Nuria. This platform aims to mine and integrate data from different online databases and to create structured review articles of a specific rare disease. And the structured reviews are presented as knowledge graphs, where the concepts are the nodes and the relations between these concepts are the edges. So this is what we've done in this project. We use this Bio-Knowledge Reviewer library together with the computational drug repurposing approach to create an automated drug repurposing workflow to find novel drug-target interactions to alleviate the symptoms of a person suffering from a specific rare disease. So for our use case, this would mean that we would input a specific rare disease, so Huntington disease. We would also input a symptom, in this case, chorea, which is the involuntary movements. And then this workflow outputs a list of candidate drugs that could help alleviate the symptoms of this specific disease, this specific person. I will explain the mechanism behind this workflow in the upcoming slides. So this is the total workflow. It seems like a lot, but I will go over every step with you in the upcoming slides. So first, I will give a general overview of how this workflow works. So first, the idea is to construct a knowledge graph. It's done in the top. The idea is to then predict the links between all the drugs and the genes of interest. And the genes of interest are the genes that are associated with this symptom chorea. This is a link prediction problem, and we predicted these links using supervised machine learning model. And then in order to get like training data and prediction data to be used by this machine learning model, we needed to embed this graph. And we did this in step 2. We did this using RDF2Vec. I will explain this later. And then in step 3, we use these graph embeddings to get prediction and training data for this machine learning model. I will now go over every step in even more detail. So first, the graph construction. We use this Bio-Knowledge Reviewer library to get information from Monarch. Monarch is an open science effort that aims to integrate genotype-phenotype data from many sources and species. So the idea is to input a disease ID, a symptom, and then get a subnetwork from Monarch. And this has different entities. We then added drugs from DGIDB. This is a web resource that provides information on drug-gene interactions from publications, databases, and other web resources. We obtain different interaction types like antagonist, antibody, suppressor, and inducer, but it's better to get, we categorize these into the direction of the interaction type, so inhibits, activates, and regulates, rather than the interaction types themselves. So we have edges with 3 categories: inhibits, activates, and regulates. And then in this project, we used a biophysical drug repurposing approach. And this has the hypothesis that drugs with similar compound structure have shared biological activity. So in order to, we need edges between the drugs that indicate that drugs are similar. And we did this using the Tanimoto coefficient. And the Tanimoto coefficient looks at the compound structure of the drug and then gives a score of how similar these compound structures of the drugs are. So we obtain one score for between every possible pair of drugs in this graph. This will lead to a large amount of scores. So we need a way to find only the most similar edges between these drugs. So we did this by only keeping the 10 most similar edges for every drug in this graph. And we did this using the k-nearest neighbor algorithm. And then we added these top 10 most similar edges to the knowledge graph and added the label similar to. We then transformed this into a RDF graph where the nodes and edges are the uniform resource identifiers. And then before embedding the graph, we shortly removed these edges between the drugs and genes. This is because when embedding, if you keep these edges for the embedding, all the drug embeddings will be really similar to the gene embeddings these drugs directly interact with. So only for the embedding step, we see it as two separate graphs, and then afterwards we would add like if the graph is known, yes or no. So this is for our use case. The graph, you see that there are 7 entities, drug, gene, variant, genotype, phenotype, disease, and pathway. And you see the different relationships between these entities. And the number in blue are the amount of nodes in each entity category. So now the embedding, we have this RDF graph. We need to transform it into vectors to be able to be used by the machine learning model. We did this using RDF2Vec. This is a mechanism similar to Word2Vec. So it uses random walks over the graph to create like sequences of entities and relationships, and then inputs these sequences into a skip-gram model. This is a neural network model with one hidden layer, and it outputs one vector for every entity in the drug. And because we are only interested in the drugs and genes, we only selected the drugs and gene vectors. And the idea is that structurally and semantically similar drugs and genes are closer to each other in the embedding space. So now we have a vector for every drug and gene in our graph. Because we want to predict the edges between these nodes and not we don't know how to predict these nodes themselves, we needed a way to like create edge representations out of these vectors. So we did this by fusing these drug and gene embeddings using the Hadamard function. So we now obtain one vector for every drug-gene pair, every drug-gene combination, and now we add the interaction type. So if it's known, then in which direction it has, and if it's unknown, we state it unknown. So out of all these edges, we need to find prediction and training data for our model. So the prediction data are just all unknown drug-gene edges of interest. So all known drug-gene interaction pairs that are associated with the symptom chorea in our case. And the training data for the positive, we need positive and negative samples because we train a supervised machine learning model. And the positive samples are just all the known drug-gene interaction pairs. And the negative samples are like a subset of the unknown pairs that are not in the prediction data. Not all negative samples are used, as this would lead to class imbalance. There are several ways to deal with this class imbalance, and we chose a method called downsampling, which means that negative samples are randomly selected from the unknown interactions that are not in the prediction data, with the sample size equivalent to the class in a positive set with the largest amount of interactions. And I will explain it in the next step. So for our use case, Huntington disease with symptom chorea, we had 1,352 drugs and 284 genes. This ended up with almost 400,000 edge representations, of which only 1,753 were known, so most of them were unknown. Out of these unknown edges, yeah, this amount was were the interaction pairs of interest. So this is the prediction data. And then the training data, the positive samples are these known edges, which in our case has 1300 regulates edges, almost 400 inhibits edges, and 60 activates edges. And then for the negative sample, we took 1300 random unknown interaction pairs, as the 1300 is the amount of the largest class in the positive sample, so the regulates edges. Okay, we now have the prediction and training data. Next step is the supervised machine learning model. We use the XGBoost classifier. This stands for extreme gradient boosting. This model has lots of hyperparameters, and the values of these hyperparameters are optimized by randomly trying out different combinations from a pre-specified set. And then for each combination of these for each combination, we assess the model using repeated stratified tenfold cross-validation together with an F1 score. And then we use the best find hyperparameters for the prediction task. So we predicted on the prediction data, and we obtained novel drug-gene interactions, so whether the interaction is positive or negative. And in case it's positive, we also predicted the interaction type, so inhibits, regulates, or activates. And we also obtained a confidence interval, so how well was this prediction? For our use case, these were the best find hyperparameters, which resulted in an F1 score of 0.867. So now we have one link between every drug and gene of interest. We have the prediction interaction type and the score. The first step is to remove all the edges that are predicted to have class none, which means no interaction. So it's not a positive interaction, so we remove these edges. So now we have all the drugs that are predicted to have a positive interaction with our gene of interest. This is the goal of this project. However, this is a really long list of positive drugs. So we need a way to only find the best predictions and the best, and then rank it in some way. So we decided to only find the best predictions by removing all the predictions that have a confidence score lower than 0.9. So only keep the best predictions. And then out of these best predictions, look at group by drugs and look at the amount of interactions these drugs have with all the genes. So when a drug has more interactions with genes that cause chorea, we say this drug is ranked higher than a drug that only has one interaction with this gene. So in this case, drug 2 only has one interaction, so it will be ranked lower than drug 5, which has 2 interactions. In case that drugs have the same amount of interactions, we sum this confidence score for this specific case and then rank the drug as a higher confidence score, higher than the other drug. So these are our results for our use case. These are the top 10 ranked drugs for Huntington disease with symptom chorea. We manually explored the top 3 ranked drugs, and we saw that all 3 could be linked to other neurodegenerative disorders, which is a good thing. So now the idea is that lab researchers can test if this can actually help alleviate the symptoms, and it's not like this will help, but it's like for a next step for persons to see if this actually works. So the aim of this project was to use the bio-knowledge review library together with a computational drug repurposing approach to find novel drug target interactions. We built an automated drug repurposing workflow, as explained in this presentation. This uses graph-based analysis methods together with network analysis methods and machine learning to create a ranked list of candidate compounds. There could be some improvements for this project. So first of all, find better negative samples. We now use all the interactions that are unknown as negative samples. However, there could be some negative samples that are unknown, but could be possible, just unknown yet. So it would be better to find experimentally validated negative samples, and this could be done using a database like CAMBL, but it's a bit of work. Next improvement would be to put more effort into creating a more elaborate knowledge graph. So, for example, add more seeds in the monarch graph. For example, diseases that are linked to our disease of interest, or all the genes that are associated with this specific rare disease. Next, some more information about the drugs could be added. So now we base the similarity of drugs only on how similar the compound structure is. Yeah, you could also add like side effects of the drugs or the diseases the drugs help with for a better finding how similar the drugs are. And finally, we kept the FAIR principles in mind. However, it would be an improvement to store the graph in like a platform like Neo4j to enable more, to enable to see it, how the graph actually is. And yeah, we try to do it, but it's still a work in progress. However, using this workflow as it is already gives results that is interesting to test experimentally. For our use case, there are some drugs that are linked to other neurodegenerative disorders, which means this is a good, the workflow could be used in practice. So happy person. We already took some steps towards turning this workflow into a web app. The idea is that researchers with no technical expertise can just input a disease and a symptom, and then the workflow is triggered, as I mentioned today, and then outputs a ranked list of candidate drugs. I did this in the web platform Flask, the Python Flask, but it still needs some improvements, and someone is now currently trying to improve this web app so it can actually be used by other people. Yeah, it's a long video, I won't do it. Are there any questions?
Yes, any question, please raise your hand.
Ooh.
Hi, thank you for your presentation. I saw that you used Huntington disease as a use case, and I see that it's really often used as a use case because it's a well-described disease. There are a lot of known drugs and genes related to it. But how does your model actually perform with other rare diseases? Like you showed the F1 score of 0.86 or something, but what is the average, for instance, of other diseases?
Yeah, we actually tried it on another rare disease, SCA1. I'm not really sure what the F1 score was of this disease, but it is possible to try with different rare diseases. And also when using my app eventually, I also run it on Alzheimer's disease, for example. So it's really easy to run it on different diseases. But I don't have the score for different diseases.
No, yeah, I think it would be nice for the general statistics to see the performance of your model if you have those summaries.
Yeah, that's a good suggestion. Thank you.
Yeah, so I have a question. This pertains more to the evaluation procedure.
Yeah.
So I saw that you are using a 10-fold cross-validation for doing that. However, it's also very well known that there's lots of near duplication in the drug target pairs. So for instance, there are many isomers or isoforms are often recognized in the drug target pairing, or they're very, very similar drugs that are shown to do the binding. And therefore, you're sort of, when you use these data sets as is, you're overrepresenting a lot of the same kinds of interactions. So I was sort of wondering whether you took any steps to make sure that your model was more generalizable in the sense that you don't have so many very similar pairs in the data set. And this is, other work have shown that this is really important to learn a general model and not be over-reliant on seeing the same predictions, because your prediction success will increase dramatically by the fact that there are so many as very similar pairs.
Yeah, we didn't take this into account in this current model, but it's good for future work. Yeah.
Okay,
Thank you for mentioning it.
thanks.
Was there a hand in the back or? Yeah, I think.
He also had a
Oh,
Sorry.
right, and also back
Yeah.
there.
Thank you. Thank you for presentation. My question is, maybe I missed this part, but how did you find the drug-drug similarities? And I think you only keep the top 10, I mean, similar drugs or something for a specific drug, right?
Yeah.
In the workflow. Yeah, maybe I missed this part, but can you explain more about like what is the procedure that you do the filtering?
You mean this step, right?
Yeah.
Almost there.
Yeah.
This one.
Yeah, thank you.
Well, what's exactly the question? Like,
Yeah, I don't, like, can you explain detailedly like how you, like, let's say keep the top 10, like what is the procedure, like how you do this
yeah, we use
process?
this K nearest neighbor algorithm. So the top 10 best predictions for every drug.
Okay. Okay. And then my second question, because it's a automate workflow, right?
Yeah.
So like now you are only like the RDF2vec is like the default or the only method that you are gonna using for this embeddings. Have you considered other methods can do that? Because I think it's a knowledge graph and you can use quite a lot of methods to do the embeddings. And I think it would be better if you can include multiples and do a comparisons of different models. And yeah, to see the score at the end, that may
Yeah,
can be
I
better. I don't know.
actually read a paper somewhere that RDF2vec was, someone tested out different methods and RDF2vec came out as the best method for this. So this is why I chose RDF2vec.
Okay, okay.
Yeah.
Thanks.
Right, you can go ahead.
Hi. Thank you very much for the talk. Very cool concept. So my question is kind of similar to the first two. It's a two-part question. So you mentioned that the amount of positive samples you used for training was around 1,000 something. Would you say that it is enough to properly train a model? And is this a common challenge for rare diseases? And is there a concern about overfitting models?
Yeah, you mean that in rare diseases, it's possible you don't have that many positive.
Yeah.
Yeah, I think, I'm not sure how the amount of positive interactions affect this model, but yeah, I assume if you have less positive edges, the model will not perform that well. But I'm not sure what the optimum number is or the minimum number. I'm not sure.
Okay, thanks.